name is Laura Niedernhofer. I'm a professor of biochemistry, molecular biology, and biophysics at the University of Minnesota. I direct the Institute on the Biology of Aging and Metabolism, and I've been working in the field of cellular senescence for over a decade. I'm very proud and pleased to have 2 grants within the SenNet Consortium, one to map senescent cells in human liver and adipose tissue, and a second to help map senescent cells in lung, muscle, brain, adipose tissue, and liver of mice across the health or the lifespan of both organisms. I'm currently on the steering committee and have been nominated to be part of the executive committee that interacts directly with NIH to help lead the SenNet Consortium.
What drew you to the field of senescence research?
I got attracted to studying cellular senescence because my background is in DNA damage and repair. And when you study DNA damage, you think of that as being extremely stochastic. So I was part of a group in the Netherlands during my postdoctoral training that really linked DNA damage, not only obviously to cancer, but now also to aging biology. But we could never really understand how stochastic DNA damage that happens randomly in cells could give you such a really common phenotype of aging biology. And it turns out that DNA damage is one of the most potent drivers of cellular senescence. So that was truly an aha moment in my own personal research. If DNA damage that occurs in your body through normal metabolism drives cellular senescence, that's what allows aging biology to happen. And so this is how we linked senescent cells to so many age-related diseases, as well as just a causal factor in aging biology.
Please give a brief overview of the research you and your team perform.
The research that we are doing at the University of Minnesota for SenNet, as well as for other funding opportunities from both industry as well as NIH, is really to understand the fundamental biology of senescent cells and how they are linked to other hallmarks of aging. Things that go wrong in mitochondria, with autophagy, with protein regulation, DNA damage. So we really want to understand what drives cellular senescence and what then senescent cells do to an organ and an organism. We do this in many, many different age-related diseases using various mouse models of DNA repair deficiency that drives type 2 diabetes or heart disease or dementia. And we're really, really excited about this opportunity because of the new emergence of these state-of-the-art technologies that allow you to do all of these analyses in tissue sections, getting both transcriptomic and proteomic data at the same time. And so I think this is going to be absolutely imperative for converting what SenNet has the capability of doing into transforming healthcare, where we can actually measure senescent cells, have biomarkers of them, and turn that into therapeutic opportunities.
What would you tell students and junior investigators who want to research senescence?
I think young individuals really need to know that aging biology is the biggest biomedical problem on our planet right now. And this is globally. And cellular senescence is giving us an opportunity to really interfere with this aging biology. I think it's some of the most exciting and intriguing biomedical topic you could possibly tackle right now. There's so much to learn. There's so many emerging technologies that can be applied. It requires incredible bioinformatics skills, which are going to be absolutely essential to anybody, I think, looking towards a research career. So I think this is like, SenNet is the perfect opportunity to get all the skill sets that you need to become a brilliant scientist, as well as potentially a physician scientist.
What are your thoughts on SenNet as a community and resource?
I believe the SenNet Consortium is absolutely essential for tackling senescent cell biology. If we all went off and did this in our own little laboratories or corner of the universe, we would not reach consensus. We would not make progress fast enough. I think there's such incredible potential here for creating technology tools, bioinformatic tools that will allow us to understand how cells interact with one another, impact one another, and therefore impact human disease. And I think ultimately everything that we can build through the SenNet Consortium, working together with common wisdom and different backgrounds, is really going to be highly impactful, well beyond just the study of cellular senescence. It can teach us about neural networks or how cancers interfere with human physiology. I just think the opportunities here for impacting translational research are enormous. I would like to add that I am extremely grateful to the wisdom of NIH for supporting this SenNet Consortium. I'm thrilled that it is part of a common fund. And I think just the entire topic of geroscience, which is the study of fundamental aging biology, which is the greatest risk factor for virtually every chronic disease, is critically important. And the more buy-in that we get from NIH and the more outreach we can do through SenNet is going to be absolutely critical for having impact. I do want to acknowledge that in January 2024, Judy Campisi, who is really one of the pioneers in senescent cell biology, tragically passed away, and we all miss her desperately. My personal feeling is that there someday will be a Nobel Prize for senescent cells, and I would have wished this would award to Judy because she has just been such a champion in the field. So we are all missing her greatly right now.